Before we start the podcast, we wanted to let you know about WestOS. If you're a teacher or pupil at a school in Scotland, then you now have access to hundreds of quality assured video lessons created by teachers to support your remote learning. You should be able to find WestOS within the app library of Glow, where you can add the tile to your own personal launchpad. Click on the tile and you'll have immediate access to stream lessons in every area of the Scottish curriculum, with video lessons already available for biology at National 5, Higher and Advanced Higher. Now let's get on with the podcast. This podcast is sponsored by Lecky textbooks, revision guides and practice papers, everything you need to learn, review and prepare for your senior phase assessments. Browse the books at www.leckyscotland.co.uk and get 40% off using the discount code LeckyPodcast40. We're thrilled by the overwhelming support for our episodes featuring top-notch experts delving into various biological realms. Thanks to the feedback from higher biology students, we're launching shorter, punchy revision episodes. Get ready for quick summaries of key areas to turbocharge your exam prep. These short episodes can support your exam preparation and are not intended to replace other important aspects of your revision. Today, we're diving into metabolic pathways, a key concept that underpins life itself. Metabolism is the sum of all biochemical processes in an organism, involving a complex web of enzyme-controlled reactions. Understanding these pathways is essential, not just for the exam, but for grasping how living systems function. So let's break it down. So what are metabolic pathways? Well, metabolic pathways are the sequences of enzyme-controlled reactions in a cell, and they can be categorised into two main types: anabolic pathways and catabolic pathways. Anabolic pathways build large molecules from smaller ones and require energy. An example might be protein synthesis from amino acids. Catabolic pathways break down large molecules into smaller ones, releasing energy. And an example of a catabolic pathway might be respiration, breaking down glucose to release ATP. Metabolic pathways can include both reversible and irreversible steps, as well as alternative routes, which allow cells to be flexible and efficient in their metabolic activities. The plasma membrane plays a crucial role in metabolism, and it's made up of phospholipids and proteins. It follows something called the fluid mosaic model. The fluid mosaic model describes the cell membrane as a flexible, dynamic structure. It's made of a phospholipid bilayer with hydrophilic heads facing outwards and hydrophobic tails facing inwards, creating a selective barrier. Proteins are embedded throughout, acting as channels, receptors, or enzymes, while cholesterol adds stability and carbohydrate help with cell recognition. The term fluid refers to the movement of these components, and mosaic reflects the variety of molecules within it. This structure allows the membrane to be selectively permeable, controlling what enters and exits the cell. Now, those proteins in the membrane can act as pores, pumps, or enzymes. The pores allow metabolites to diffuse in and out, while the pumps move substances via active transport, which, remember, requires energy. And enzymes catalyse metabolic reactions directly in the membrane. Now, enzymes are biological catalysts that speed up metabolic reactions by lowering the activation energy. They operate through something called the induced fit model, where the active site changes shape to bind the substrate more effectively. Metabolic pathways are controlled by the presence or absence of particular enzymes and the regulation of the rate of reaction of key enzymes. Enzymes catalyse metabolic pathways by reducing activation energy. Now, that's the energy required by a reaction to proceed. The energy is required to break bonds in the reactant molecules. The active site of an enzyme is the location where substrate molecules bind, allowing the chemical reaction to take place. Induced fit occurs when the active site changes shape to better fit the substrate after the substrate binds. That ensures that the enzyme and substrate are in close contact to increase the chance of a reaction taking place. Now, an enzyme has an affinity or a chemical attraction for its substrate. Products have a low affinity for the active site of the enzyme and are therefore released from the active site after the reaction takes place. Most metabolic reactions are reversible, and the presence of a substrate or the removal of a product will drive a sequence of reactions in a particular direction. As the concentration of substrate increases, the rate of an enzyme-controlled reaction also increases up to a point at which the rate of reaction remains constant. At that point, some other factor will be limiting the rate of reaction, possibly enzyme concentration or something else. The shape of the active site of the enzyme determines the orientation of the reactants, and that's vital when there are two or more substrate molecules to ensure that they're held together in a way that the reaction between them will take place. Enzymes don't always work at full capacity. Sometimes their activity is deliberately slowed down or even stopped, and this is where enzyme inhibitors come into play. These molecules regulate metabolic pathways by decreasing enzyme activity, ensuring that reactions only occur when necessary. Metabolic pathways are carefully controlled by enzyme regulation, which includes the following three inhibition mechanisms. Competitive inhibition involves inhibitors with a similar shape to the substrate which bind to the active site of the enzyme. Competitive inhibitors prevent the substrate from binding to the active site of the enzyme, thereby reducing the rate of the enzyme-controlled reaction. The effects of competitive inhibitors are reversible, so increasing the substrate concentration increases the rate of the reaction by increasing the chances of active sites being occupied by substrate molecules rather than the inhibitor molecules. Non-competitive inhibition involves inhibitors binding to a part of the enzyme molecule other than the active site, and that part of the enzyme of a non-competitive inhibitor is known as the allosteric site. When a non-competitive inhibitor binds to the allosteric site, it alters the shape of the active site. Now, the effect of non-competitive inhibitors are permanent, and its effects cannot be reversed nor reduced by increasing substrate concentration. And thirdly, feedback inhibition occurs when the end product of the metabolic pathway reaches a critical concentration, and the end product then inhibits an earlier enzyme, blocking the pathway and so preventing further synthesis of the end product. This is really useful in terms of preventing overproduction. And these controls ensure that metabolic reactions only occur when needed, conserving energy and resources. So metabolic pathways are more than just a topic for the exam. They're the foundation of life itself. Without the precise control of enzyme activity, life would not be able to function efficiently. So next time you eat, exercise, or even just breathe, remember that your cells are carrying out such complex metabolic reactions in every moment. So hit subscribe on whatever platform you get your podcasts so you don't miss the rest of our short summary episodes nor our regular episodes with leading experts. And remember, this quick summary is intended as just one element of your broader revision plan. There are lots of resources available via Glow such as Scholar or WestOS, as well as BBC Bitesize. Or why not check out one of Lecky's great revision books and use the code LeckyPodcast40 to get 40% off on their website www.leckyscotland.co.uk.